the organizers for the opportunity for me to be here and present you what we have been doing the last 2.5 years between my group, the group of Nikos Daskalakis, who's there in the audience, and the group of Brigitte Startset, that is in the University of Aarhus. So basically, our interest are the mitochondria, and specifically how the mitochondria, the nucleus, integrate their functions. So we all know the mitochondria. They're very interesting and very intricate organelles that are very important, and they actually control the energy production in the cell. But they are not only that. They do a lot of more things. For people that are more passionate about mitochondria, like myself, they do other than ATP production. They control the urea cycle. They contribute to calcium homeostasis. They regulate cell death. And to do all this, they have to coordinate their actions, and they have to coordinate their actions with great dynamicity. And to do that, there is only one way to do it in the cell. It is through signaling. So basically, it is well accepted now that mitochondria are basically signaling hubs, and they can integrate their functions with other organelles through signaling events. Now, which organelles are interested in this kind of communication? Of course, it is very well studied, the communication between mitochondria and the endoplasmic reticulum, but a little bit less surprisingly, less studied is the communication between mitochondria and the nucleus. So now, mitochondria and the nucleus, they have plenty of time. They had 2 billion years to develop a language. And basically, this language goes two ways. From one way, we have the nucleus that controls basically the production of virtually all the mitochondrial proteins, and it can actually send information to the mitochondria through these proteins. So the nucleus can actually, through an anterograde signaling pathway, it can actually regulate the function of mitochondria, the production of the mitochondrial DNA, the mitochondrial biogenesis, and other stuff. In the meantime, the mitochondria, however, they need also to communicate their needs to the nucleus. And how they do that? Well, they do it through what is called the retrograde signaling, where the mitochondria, they send information to the nucleus, and they send a bunch of information. And this information is basically saying to the nucleus in real time what are the needs of the mitochondria and what is the status of the mitochondria. Am I producing enough ATP? Is there a stress? Is there something happening in my respiratory or metabolic status? So now, how they do that? How this communication, it does this? And of course, now we are called to go interpret and decode the language that was generated, that it had 2 billion of years of evolution. There is another problem though. This language, it comes through two levels. The first component of this language between the mitochondria and the nucleus is a soluble part. So basically are the molecules that the mitochondria exchange with the nucleus, and those basically, they're very simple molecules. So they're basically the words that they have been using, these two organelles, are very common. So that creates a big problem, and the problem is that how we are going to learn a language and understand what this language is meaning between two organelles when the same words are used exactly in the same moment, exactly in the same room. So imagine that all of us, we are discussing, and we have 10 words to make discussions. How should we know what two specific individuals are saying within a complex environment? So we were thinking about this problem with Nikos and Brigitte, and we thought that the best way to understand is spy on them. But you cannot spy unless you isolate the components. So what we thought it would be a great idea to actually generate an environment where we can put inside this environment mitochondria and nuclei, fill the environment with spies, and then stay there and listen to what they say. And then at the end of the day, go and see what is the functional significance. So Brigitte, she's basically a material scientist. What she did, she developed basically a matrix gel where we can put inside mitochondria and nuclei, and they are both working. That was the challenge. Then my group, we are the spies. So basically, we are the signaling people. So our job was to implement both the matrices and the organelles with sensors that they can measure basically the words that the two organelles they were exchanging. And then Nikos was coming there to give a meaning to all this by looking at the transcriptional regulation that these events they might regulate. So now, thanks to this process, the goal or our hope is that we are going to have a mitochondrion sending an information that we will measure to a nucleus, and the nucleus will give a response that Nikos will manage to actually understand. So we studied, of course, this project. We studied it, of course, with the matrices. And as you can see, Brigitte, the group of Brigitte, they tested some of the matrices, some hydrogels. And we focused on the hydrogel on the PEG, basically 500, because it has a high stiffness, but not high enough to become problematic for the FRET experiments, for the life organelle imaging experiments, but it still gives some stiffness enough to that it resembles a tissue. So basically, as you can see, we incorporated mitochondria, and you can see that the mitochondria in the gel, they are fine. This is a marker for outer mitochondrial membrane, but most importantly, both the same day of the incorporation, encapsulation of the mitochondria, and the day after, the mitochondria were energetically active because they were managing to uptake basically what is a dye that detects that is uptaken thanks to the membrane potential. Next, Brigitte went on and she did analysis of the ATP production of the mitochondria. So we had mitochondria in what is called mitochondrial respiration buffer together with substrates, in this case succinate, and then we will give or not give ADP. And as you can see from this slide here, basically we managed to keep production of ATP in all the matrices that we tested, and also of course in solution. And sometimes, surprisingly, in the 500, in the PEG 500, which was our choice, our material of choice, it actually, the mitochondria, they managed to stay alive for some days, which is quite surprising to us, and we are looking into it. So we were able to find this matricel that actually managed to have the mitochondria nice and respiring, so we decided to go on and look at the nuclei. And as you can see, again, the same story. We isolated the nuclei. We tested like 10 protocols to find the correct protocol for isolating nuclei, because they have to be clean of mitochondria, of course, and they have to be very well tight and not losing structure. And as you can see here, basically those are the nuclei in a matricel. What was interesting, it was okay, we had a little bit of calexin, but that is normal. It is expected because the mitochondria, the nucleus, the nuclear envelope is an extension of the ER. But we also, our nuclei, they still contain nuclear bodies, which are dynamic organelles, showing that the nuclei, they are in very good health. And that was also, of course, demonstrated when we did experiments by looking at the production of RNA in this encapsulated nuclei. And as you can see, up to 4 hours after encapsulation, we managed to see both normal production of, spontaneous production of RNA, but also when we did an experiment where the cells were pretreated with lipopolysaccharides to activate the the transcription factor NF-κB, and we followed then the production of TNF-alpha, we were able to see once more that encapsulated nuclei, they were very nice and they were producing RNA. So the next is, are they communicating? Is there something that we can say? Is there a principle that we can say that there is communication? And to do that, we took both nuclei and mitochondria. We put them in a matricel, but this time what we did, we actually implemented the nucleus with a sensor for calcium. So the idea was that if the calcium is a signal that the mitochondria can actually use to communicate with the nucleus, the presence of mitochondria should change something in the calcium of the nucleus. So, and that was indeed the experiment. As you can see here, this is the dark line represents a FRET life organelle imaging, where basically we could see that the calcium, the nuclei responded when they were alone. They responded very nicely to calcium. But when we added mitochondria to the equation, the signature of the calcium signals, it completely changed. Now this demonstrates that mitochondria can actually change a signal into the nucleus, a nuclear signal. Of course, it has a biological meaning, a potential biological meaning, because we know that the calcium waves within the nucleus regulate transcription, and therefore potentially we are going to be able to tune up or tune down transcription through mitochondria into and through calcium. Nikos is actually now analyzing the RNA-seq experiments from this. So we talked about this modality, the modality of communication between mitochondria and nucleus through molecules, but there is another way for these organelles actually to communicate. And actually is the establishment, this is the least studied way, is the establishment of mitochondrial nuclear contacts, direct contacts. So it is well known that mitochondria, when they feel stressed, they go away from the periphery of the cell and they go and accumulate near to the nucleus. But there are many open questions. We do not know why they do that. We do not know which are the proteins through which they are doing that. And most importantly, we do not know what is the information that is being exchanged. It's very difficult to study this because you don't get many mitochondria that go to the nucleus. So we decided to take a different approach, like an approach that nobody else will fund if not Human Frontiers. So what we did, we basically generated this tool here, which we call mitostids. Those are the mitochondria in green, and you can see the dark space, which is basically the nuclei. And as you can see, as we go on and the mitostids is expressing, the mitochondria, they are kindly invited to go and make a visit to the nucleus. And as you can see, while the time is going on, more and more mitochondria, they attach themselves to the nucleus, and this has a big penetrance. And in a very little time, what we get, probably in the first 8 hours of this experiment, we get a large population of mitochondria that they went and they touched the nucleus. Now we generated mutants, as you can see here. This is a mutant mitostids that mitochondria do not go to the nucleus, and this is the effect, basically, of all mitochondria being attached to the nucleus. So now we have a tool. Of course, we made sure before going on to do functional experiments that basically the mitochondria that are at the nucleus through the mitostids, they're perfectly healthy and they're perfectly functional. There are plenty of data that I'm happy to talk about, but I cannot present here due to time. And basically then, now we have a tool. Now we can actually induce mitochondrial nuclear, we can force the two organelles to discuss, and we can listen to them. So the first experiment we did, it was basically a signaling experiment. And the signaling experiment was very simple. We had a sensor, a threat sensor for PK activity that was targeted to the outer mitochondrial membrane, OMM-ACAR4, and the same sensor that was targeted to the nuclear envelope from the outside part. So now let's see what happens when mitochondria, they move near. So in this the first up on the left, what you can see is that the stimuli is that a small stimulus at the beginning of this experiment, basically the stimulus was production of cyclic AMP, was unable to create any pKa signaling at the outer mitochondrial membrane. So basically, when the mitochondria, they were free. When we took the mitochondria and we brought them at the nuclear envelope, once more, the same stimulus was unable to generate any reaction, which we found that was interesting. So then we went back to the nuclear envelope and we looked at the same sensor, but from the other side. Now what the nucleus is responding to the stimulus. And as you can see, once more, when the mitochondria were in the periphery, there was no response at the nucleus. But things changed when the mitochondria went near to the nucleus. So when we established the nuclear mitochondrial communication, suddenly what we found is that the signals are not anymore silent, but that spot where the mitochondrion was touching the nucleus, it actually generated a signal. I think this is very important because what happens basically when a mitochondrion activates a kinase, what we do, we basically open a whole new world because the kinases, they are very efficient in phosphorylating stuff. Therefore, there we potentially have an amplification of a cascade. So now, how this becomes function. In order to understand that, of course, we did the RNA-seq experiments, an RNA-seq experiment where we looked at the transcription when healthy, normal mitochondria were far away or they were touching the nucleus. And the group of Nikos analyzed the data, and we found that by simply moving perfectly normal mitochondria from the periphery onto the nucleus, transcription changes. But one of the most important and striking things, it was this gene. So basically, you simply need to move mitochondria from the periphery of the cell to the nucleus to upregulate consistently the biggest predictor of Alzheimer's disease, ApoE. So what we decided to do, of course, we didn't believe that was true, and we actually confirmed it also with Western blotting. So actually, indeed, by simply moving mitochondria, we can actually change the transcription, generate small hotspots of signaling, and also generate transcriptional signatures that they were unexpected. Now, it is relevant because we know that in many pathologies, Alzheimer's disease indeed, the mitochondria move to the periphery of the nucleus and they establish connection. So that was my last slide. We had many conclusions during these first years of the 2 years of the award, and thanks to basically the two beautiful collaborators. This is my team in Pavia. We are split between Pavia and Padova, and Doruk, he's the person that is doing all the experiments. And then this is the team of Nikos, and this is the team of Brigitte. I have to say that one of the most important things that we found, thanks to the Human Frontiers, it was the ability to actually take some time and meet each other. So I was able to visit Nikos for 15 days, for a month last year. It was amazing to exchange basically experiences with people that they do different things. And then I have to say that Catherine and Doruk, when Catherine visited my laboratory, she came for 2 weeks, and then after a while she said, you know what, I'm going to stay for 2 months. And they actually finished the manuscript. So thanks to the Human Frontiers once more for the support, and you for your attention.